Welcome to Believe in Better, Twist Bioscience's workshop at AGBT. My name is Chris Thorne, Senior Manager of Field Marketing at Twist. Today, we're going to be hearing from two of Twist's customers on how they're using our technology to reimagine what is possible for their research. But to begin, I'd like to welcome our CEO, Emily Leproust, who will give a short update from Twist.
Good afternoon. My name is Emily Leproust. I am the CEO of Twist Bioscience. I'd like to welcome you to our AGBT2021 workshop. First, I'd like to say that I would be making some forward-looking statement. I don't think I can begin a presentation at a virtual AGBT without acknowledging that a lot has changed this last year. When we last gathered in Florida in 2020, no one knew how different our life would be 1 year later, but one thing has not changed, and that is science does not stop. And the fact that it has not stopped means that there are more tests, better treatment, and vaccines that are now available for addressing COVID-19. And Twist has not stopped either. In 2020, we, like many others in the biotechnology and research area, have shifted our focus to ask how we could help the global pandemic. And we've done three things. So first, we've launched NGS target panels for the detection and characterization of SARS-CoV-2. And as we've seen with the emergence of new variants, being able to sequence the entire virus is a great complement to the RT-PCR test of saying yes or no, do I have COVID? And so with Twist best-in-class target enrichment technology, we have enabled ongoing environmental surveillance and highly sensitive identification of new mutants. In addition, we've also used our rapid DNA manufacturing capabilities to develop fully synthetic RNA control for the verification and validation of SARS-CoV-2 tests. Basically, we have COVID in a tube. It's a full 29 kb RNA sequence cut in 5 locations, so it's safe. You can brush your teeth with it, don't do it, but you could. And that replaces the attenuated virus that used to be used as a control, and that is quite dangerous. So we're very pleased to say that we've been building a pipeline of many mutants that have emerged to enable the development and routine verifications that diagnostic tests work. And then last, in COVID, we've worked on antibodies. And in March 2020, we've announced a partnership with Vanderbilt University. They use thousands of Twist genes to discover antibodies, and 6 of their antibodies are now in clinical trials with AstraZeneca. And at Twist, we've used our own platform to develop our own antibodies very fast, in 6 weeks, and those have been verified by independent studies to be highly neutralizing against SARS-CoV-2. So we're very pleased to aid in the battle against the global pandemic. But of course, science is not stopping, and it's moving ahead in other areas, and at Twist, the same is true. So our own end-to-end solution for target methylation is now available off the shelf, and it combines its gentle enzymatic conversion with the Twist target enrichment system, and those two things together provide exceptional performance. Here on the right, you can see how with the enzymatic conversion method, you get a more even coverage with hypo and hyper methylated area, and a lot more information is available at our booth. In addition, today you'll be hearing from our customer Regeneron and their application of a panel for genotyping by sequencing. And so by combining their expertise in genomic discovery and statistical power with the Twist expertise in panel design, as well as our innovative target enrichment probes and reagent, together we have developed a flexible, scalable, and unbiased population genetic screening panel that has allowed Regeneron to streamline their workflow, cut down their timelines, and cut down their cost, and as well produce richer data. So today you'll be hearing how, using this panel, they are moving from the legacy microarray-based projects that they've been using to NGS genotyping by sequencing, and enable economical and improved association studies. In addition, I'm pleased to say that the same approach is broadly available to our customers who want their own content, and that is available either as a standalone panel that we can develop with you, or that will be available as a spec-in that you can spec into your existing panel. So if you're doing microarray analysis, you don't have to wait for new content, and you can switch to sequencing. So this year, our time is very brief, and I must be brief as well. And so I am delighted to be joined by two of our customers, Gonzalo Abecassis from Regeneron Genetics Centers and Kenneth Chahine from Helio Health. Our speakers will also have a short amount of time, and so they'll only give you a tease and a highlight of their presentation. But with this in mind, I'd like to invite you to join us on March 17th, where you can hear longer presentation of both of our speakers as well as other speakers in a half day of a program on the theme of cancer research. And to recreate the AGBT experience, we've set aside some really special swag for AGBT attendees. So be sure to go to our booth and register to attend. With that, thank you very much for your attention and enjoy the talks that follow.
Thanks, Emily. Now I'd like to welcome Dr. Gonzalo Abecassis, VP Chief Genomics and Data Sciences Officer at the Regeneron Genetics Center, where he oversees statistical genetics and quantitative data sciences, driving novel genomic discovery. Dr. Abecassis has co-authored more than 250 scientific papers and made key contributions to many important genomics and data sciences initiatives, including TopMed and Genes for Good. His studies and those of other scientists using his tools are enabling a better understanding of human genetic variation and its role in disease biology. Welcome, Dr. Abecassis.
Hello, really nice to be here today. I'm going to be talking to you about an approach that we've been using to really modernize and scale up genotyping. And before I do that, I wanted to take a moment to just tell you about the Regeneron Genetics Center where I work. So at the Regeneron Genetics Center, our goal is really to use human genetics to get clues about biology and about disease, and and use those clues to then design better genetic studies, eventually better medicines, better interventions. To give you a sense of scale, we're planning to sequence over 500,000 samples this year. We've sequenced more than 1.5 million samples over the past few years. And, several of these have gone on to seed drug discovery and target biology programs throughout Regeneron. Really our ambition is that we will be putting together one of the world's largest combinations of DNA information and health information. We've been doing this with many partners. You'll see many of them there on the right of the slide. As we go about our work, one big challenge is that arrays, which are a really good way to assess common genetic variation, really don't scale well to these numbers of samples. They're very convenient if you're doing modest numbers. By the time you say, let's look at 500,000 samples or 1 million samples a year, they don't keep up. And so we've been thinking about, what would it take? What would a strategy where we are both doing exome sequencing at scale and genotyping at scale look like? We had a few goals. We wanted this strategy to be at least as good as current arrays. We also wanted it to work really well across a range of ancestries and populations, not just in Europe. We also ideally would like this to simplify our process in the lab. So where we ended up was with a new sequencing-based approach that I'm going to be telling you about that's built on the same DNA capture technology that's now used for exome sequencing. And so it integrates really well in the lab. It's easy to automate. It can be carried out exactly in parallel with the exome sequencing. And it's got a few nice features that are different from standard arrays. It's tunable. So for example, in our own applications, we go for deep sequencing of the exome. We want to get every rare variant exactly right there. But we're a little more lenient in the rest of the genome. We want to capture common variation there. And so we can tune that not just between exome and genome, but we can actually tune that at the level of individual variants of interest. Another thing we thought was important to do was the way arrays were designed. They were designed for technologies where it was important to pick each variant individually. It was important for variants to be far apart from each other so they wouldn't interfere with each other's genotyping assays. That's no longer required for sequencing. We actually prefer if variants are close together and they can be sequenced with the same probe. So I'm going to show you a little bit of the approach first in words and cartoons, and then a little bit of preliminary data that shows it's working really well. So in words, we set up an algorithm that tries to capture common variation across the entire genome. The algorithm is trying to do this efficiently to both represent all available variation, but to do that with a relatively small number of targets. We've started with a set of probes that we developed in silico, and then we've gone through a series of experiments to refine and validate those in the lab. We currently have a set of about 1.4 million sites that we are targeting. We try to represent, in addition to all common variation, a few regions of interest really well, particularly variants that are known to be associated with human disease, HLA, pharmacogenetic variants, the mitochondria, and the Y. Currently, we're using this for our own work at Regeneron, and very happy if you're a Regeneron collaborator to talk about how we might apply it to your study. But we're also working with Twist to try and make this available as an off-the-shelf commercial product that anybody could use. If you look at this cartoon to try and lay out what we're doing, imagine that each of these lines is a segment of DNA, and we have both genes and coding regions here, these orange boxes, and then we have a series of variants represented by each of the circles. If we now focus on a single one of these genomes, when we carry out a traditional exome sequencing approach, we try to capture sequencing reads represented by the double-headed arrows that cover coding regions in particular. And we've now supplemented these with reads that try and capture variants outside coding regions represented by the sort of yellowish double-headed arrows. How does this work? Well, here's some real data. First, you'll see in the bottom row, current standard array, the GSA array, for example, targets about 500,000 sites. It can capture those pretty well. 98.9% of sites are called, and when they're called, the concordance with deep whole genome sequence data, about 99.9. When we look at our own experiment, you have two rows. One just using a standard variant caller that just looks at the read data, makes its own decisions. And then the second row, our recommended approach that really takes into account what's known about haplotypes in each region and tries to make even better calls. And you'll see that there we get missingness of only about 1%, 98.99% of sites are called, and concordance is also excellent at almost 99.8%. So, and we're doing this for less than the cost of the GSA array, and with less complexity in the lab, because it's done in parallel and in the same set of reactions and capture and sequencing as exome sequencing that's happening for every sample in parallel. This is a little bit of data in terms of performance. In this particular graph, you can see along the x-axis how we're doing across allele frequencies, and on the y-axis how we're doing in terms of capturing genetic variation. And you have three lines. In blue, what would happen with the GSA array. in red, what happens when we do our combined genotyping and exome sequencing experiment. And for comparison, in gray, what would happen if instead of the GSA, you went for the mega array, which is one of the most dense genotyping arrays available out there. Because it was important that we perform well, not just in Europeans, but across many populations. In the next slide, I'm going to show you performance, particularly in individuals of African ancestry. And so, as you can see, again, in blue, you have the GSA array, and then in red, genotyping by sequencing. And actually, together with genotyping by sequencing, you have the performance of the mega array in brown if you squint and look really carefully. And you can see that basically we're getting excellent capture of all common variation. And once you do imputation, doing really well down to frequencies, close to 1 in 1,000 or so. So, summary of what I've showed you, giving you examples of a sequencing-based alternative to genotyping arrays. It's not as simple as just taking existing variants and that were used in arrays and putting them in a capture reagent. We really tried to reimagine things to capture multiple variants together when that made the sequencing more efficient. We shown you that it works very well in parallel to exome sequencing, that it captures variation across the genome. Talked about how this experiment can be tailored to ensure that any high interest variants get even better quality, and shown you some data on accuracy, which has been really excellent. The other final point to make is that with a set of open source tools, you can put together a bioinformatics pipeline to analyze this data. It only adds 10 hours or less of processing time to each sample, which is really straightforward to carry out on any of the modern computing clouds, and really translates into only a very modest sort of computing cost, probably on the order of 10 cents to 20 cents, depending on the deal you have with your cloud sequencing provider, cloud computing provider. All right, some of my Regeneron colleagues that helped put this together acknowledged on the slide, and I think that's what I wanted to show you for today. Thank you so much.
Our final speaker today is Kenneth Chahine. Kenneth Chahine is the Chief Executive Officer at Helio Health, an AI-driven healthcare company focused on commercializing an early cancer detection test from a simple blood draw. Previously, Ken was an Executive Vice President and General Manager of Ancestry.com, prior to which he was CEO at Avigen. Thank you, Ken.
Thank you very much for the opportunity to present today. I'm Ken Chahine, the CEO of Helio Health, a company focused on early cancer detection screening. Not going to spend a lot of time here, but this is our senior executive team. As you can see, I came from Ancestry and launched Ancestry DNA. RF Nakuda has joined us from Amazon. Samuel Tong is our CEO for China. We have full operations in China, and he came up the ranks through Abbott, Becton Dickinson, and Kaizen, where he led Asia operations. And then Ben Euler is our Chief Operating Officer. He worked with me at Ancestry and frankly did most of the heavy lifting for things that I take credit for sometimes. Love to introduce very quickly Helio Health. I think an important way of looking at early cancer detection is, when we're sitting here in 10 years looking back, what companies were the ones that were successful and which ones weren't and why? And as you think about that, the one thing that really jumps out is the need for data to drive AI and to drive ultimately better performing algorithms. And today, when you look at the early cancer detection space, it's progressing very nicely. We're seeing some good data, but there really hasn't been the large scale amounts of data, patient data that we need. And so one of the advantages we have is having full operations in China and in the United States. We're leveraging China as a sort of discovery engine, a hypothesis generating engine for us that then can be leveraged in the US and can be leveraged globally. And so this is really important for us. And I'll share with you some of the things that we're doing right now. And I think, that is going to be ultimately the differentiator, finding partners like Illumina, who we've worked with for a long time, but also Twist, who can help us really develop the tools and work with us to develop the tools that we need to really continue to accelerate the development and production of these products is going to be very critical. We are a blood-based platform. Methylation is really our key technology that we're using. And so for a lot of the work that I was just talking about, we'll be looking at the methylome and really trying to identify combinations of markers that are really predictive in the space. We have just completed a clinical trial in China. It was almost 1,100 subjects. For liver cancer, the performance of that test was very, very good. We had stage 1 performance of over 90%. We're also doing a trial in the United States, which is a prospective trial comparing to ultrasound, which is currently the standard of care. And so far that trial is actually performing very well also. We're at a point right now where we're launching in China this year and hope to get our regulatory IVD approval in China by the end of this year. In the US, we're hoping to complete the clinical trial this year as well, and then have a launch in the second half this year as an LDT, and then in 2022 launch with FDA approval. We are a platform technology, and so we continue to push on colon, but also breast and lung cancer, where there's really tremendous opportunity and an unmet need. We have some great partnerships both in US and China. As you can see, we have multiple other clinical trials that are being run to really push the understanding and learning and also KOL development in the space. And then finally, just a note, right? The market for early cancer detection is really, really large. And it's something that we find very attractive from an economic standpoint. This slide's a little bit busy, but just wanted to give you some concrete, sort of data on some of the stuff that we're doing in China. We have multiple trials that are ongoing for liver, colon, and lung in very large scale. And this is what's going to be driving a lot of our discovery engine. And one of the questions that we get quite often is, look, we like the hypothesis, but, can you really translate the data from China into the US? And just show you a little bit of data to give you some comfort around that. We're looking at methylation markers. This is one of the methylation panels that we're using for our liver cancer test. And as you can see in the US data on the top left versus China on the right, you're seeing some very good concordance in this hypermethylated state of these markers that we're looking at. And likewise, when you look at the hypomethylated markers that we're using in our test for liver cancer, you can see some really amazing concordance. So we were very pleased to see this, and it's obviously very encouraging for the thesis of using China as a discovery engine. And ultimately, the test is the performance. And in this case, what we did was we took the developed the algorithm in the United States with US data. The China samples were 100% naive, had never been used for any part of the training set or marker development. When we applied the algorithm to the China data set, as you can see, it also performed very, very well. So this is, again, very encouraging, and we want to continue to push very hard on doing additional discovery work for colon, for breast, and for lung, because we think there's just a huge opportunity and very few people really working in that market. So to sort of wrap up, I mean, if you look at the competitive advantages for Helio, but also the way we see this market basically evolving over time, as I mentioned, data is going to win, and that's something that we're really, really focused on. I do think we need to make it simple and increase awareness. I think that a lot of people today are not getting the early cancer detection screening they need to get. A lot of this has to do with the fact that it's challenging, like a colonoscopy, et cetera. And I think moving to blood-based tests is going to be absolutely critical. I think driving in-home testing is also going to be really important. That was already happening. COVID has continued to drive that trend. And so we believe that this is going to be important to get increased adoption and also adherence. And then finally, from an economic and business standpoint, the early cancer detection market is really fantastic in the sense that it's a repeat business. And so that's something that we really want to do, is get people in our ecosystem and then make sure we're driving the annual or biannual testing that needs to happen to make sure that they're safe, and if they do have cancer, that we catch it at an early stage. So that's a little bit about Helio Health. I want to thank everyone again for the opportunity to present today.
So that brings us to the end of Twist Bioscience's workshop at AGBT. I'd like to thank all of our speakers today, as well as you, for joining us. The virtual format of AGBT has changed many things, but we still have our booth, so if you want to continue the conversation about targeted methylation sequencing or genotyping by sequencing, please stop by and talk to a member of our team. And if you complete our short survey, we'll send you some Twist swag. At our booth, you can also register for our upcoming Writing the Future of Cancer Research meeting on our brand new virtual platform. As AGBT attendees, if you register at our booth, you'll also get some extra special Writing the Future swag. So please do go check it out. Thank you all once again for joining us, and enjoy the rest of the AGBT meeting.